Hi everyone, this lab video is reviewing aseptic technique, also called aseptic transfer. But before we dive into the details of this lab, I want to review a few key terms with you that I have used in previous videos, but I want to make sure we're all crystal clear on some of the lab terminology that you are going to hear this semester. So a culture is a population of cells growing within a growth media. So when you have a broth culture that's, or like a liquid broth media that has a lot of E. coli growing in it, you have a culture of E. coli. If you have a bunch of Staphylococcus aureus growing in your broth, then you have a culture of Staphylococcus aureus. Okay, so if you have a population of cells in your medium, that's a culture. Now, in the two examples I just gave, those were pure cultures. You only have E. coli in your broth, that is a pure culture. But normally when we do this lab, we actually give you a mixed culture, which means there's more than one species within a sample. So normally with this lab, and what you're going to see on the results PDF, well, actually that might be in the streak plate. So sometimes we give you a mixed culture because then you can see the different organisms growing and that they grow differently on agar plates. But it kind of depends on the semester and the instructor. But if you have more than one species growing in a test tube or on a plate, you have a mixed culture. Now in this class, normally at the beginning of lab, I would give everyone the bacteria that they would need for that day's class. So you might have test tubes of E. coli, you might get test tubes of Salmonella or Shigella or whatever. And I would say, okay, these test tubes have bacteria growing in them. I need you to put some of that E. coli onto an agar plate. Or I need you to take some of that E. coli and inoculate this new broth media because we're going to test and see if it can metabolize this sugar. Okay, so you would be taking bacteria from a culture I give you and putting it into a new media. That process of transferring microbes from one culture to a new media is called inoculation. Okay. So again, you're taking microbes from a test tube I give you and introducing them to a sterile media so that they can grow. Now, please know your starting media may not be a broth. It could be a slant. It could be an agar plate. Typically, broth is just the easiest for me to make and to distribute to everybody. So a lot of times your starting cultures are broth cultures, but not always. They don't have to be. So when you are inoculating, you want to make sure that you don't contaminate yourself, your workspace, anybody else, right? Or the new sterile media, right? Nobody wants contamination. And so the process of transferring organisms without contaminating anything is called aseptic technique or aseptic transfer. Okay, I'll normally say aseptic technique. So let's take a quick look at some of the tools that you will be using in lab and kind of just the normal work setup when you're going to be doing aseptic technique. So this left-hand picture are various types of tools or equipment that you would use in the microbiology lab. The most commonly used tool is the inoculating loop. I'll just call it a loop from here on out. And so this top part, let me get the highlighter handy. This top portion right here that I'm highlighting, this is the handle. Okay, so that's what you would hold, much like a pencil. And the other end, okay, is the specific type of metal that forms a loop on the very end, hence why it is called a loop. And the metal that makes up this wire loop heats up really, really quick, which is important because we need to sterilize this loop quickly, but it also cools pretty quickly. That way you're not standing around forever waiting for your loop to cool down, right? Because you need it to get really hot to sterilize it. But if you then take your super hot loop and touch an E. coli cell, it's going to die, right? It can't withstand crazy heat just like we can't. So it has to be able to heat up and cool down very quickly, okay? And so that loop is what you use to touch your starting culture, okay? Whether it be a broth or a colony growing on an agar plate, or a lot of bacterial cells growing in a slant. Now, remember, I mentioned in the previous video that for agar deeps, you use an inoculating needle, and it's not like a syringe needle. It's an inoculating loop that has the loop part cut off. So this right here is the inoculating needle. So it's literally, we buy inoculating loops and we can cut off the ends so that it's just pointy. And that's what you use to stab the agar deeps. Okay, remember we said that those are important for motility and like oxygen requirement experiments. Okay, so just a quick reminder, you always want to label your media, okay? And when it comes to broth cultures, which are liquid cultures, in a test tube, you want to always label the glass test tube, not the lids, because lids come off and can be shuffled around or lost. Slants and deeps are also in test tubes, so same rules apply here. And with agar plates, you always want to label the bottom of the plate. And in the previous lab, the worksheet spells out all the things you need to include when you're labeling your media. And so again, normally you're going to have, your name, the instructor, what organism you have, and some kind of hint about the environment. And those are typically what you're going to put on a plate. And notice here that you write your information along the edge of the agar plate, because the agar plate shown here is a blood agar plate. It actually contains normally sheep blood. But a lot of times the agar plates are that more like beigeish, clearishy kind of color, like you see in this bottom picture. And so you don't want to write real big in the middle of your plate, because it's going to skew your results when you're looking at it. You're going to get distracted by your Sharpie writing on the bottom of the plate, and it might make viewing those colonies a bit more difficult. So down here, this bottom image is essentially what you all would be doing in the lab. And notice that there's a Bunsen burner. That's what we use at ACC. And not only does the Bunsen burner kill any contaminants, that it's touching, but it also creates like this bubble, this sterile bubble. Okay, so let me actually turn on my video real fast. Didn't really prepare for this in terms of appearance. So sorry, y'all. So let's pretend this is my Bunsen burner. Okay, and there's fire coming out of it. Of course, it's killing anything in that direct fire path, but the heat radiating from the flame also creates like this sterile bubble. And so you actually want to work close to that flame. Now, not so close that you're going to burn yourself or accidentally stick your loop full of bacteria in it, but you do want to work close. And that's the only real issue with this picture that I have, is that they are inoculating that test tube pretty far away from the flame. Okay, so you really do want to, I normally keep my rack right in front of the flame. That way I'm working, with a few inches separating my hand and the flame. Okay, so you do want to work within that sterilization bubble. So I am going to demo for you what's on this slide and what's spelled out in the aseptic transfer handout. Okay, so now we're going to pretend that this is not a flame, okay, that it's my starting culture. Pretend it's a broth of E. coli. This pen is going to be my inoculating loop. You hold it like a pencil, okay? Now use your imagination and pretend there is still a Bunsen burner here. The first thing you always do is you sterilize your loop, okay? I say that you flame your loop. You put it inside the flame. The wire loop will glow, okay? It heats up real fast and it turns bright red. And that's how you know it's sterilized the environment. And then you remove your loop from the flame because we need to let it cool. But during this time, we need to get the lid off of our test tube. You normally want to kind of loosen your lids in advance. And so here's where things get exciting. You want to use your pinky finger to remove the lid from your test tube. And notice I'm holding my test tube at an angle because I want to minimize the air contaminants that can gain access to my test tube. Now you want to sterilize that test tube because it's exposed to the air. So you pass your test tube through the flame, and then you insert your loop and you grab bacteria. Now at this point, you have bacteria on the end of your loop. So you don't want to move your loop much because you don't want to risk flinging bacteria across the room. So at this point, you really want to leave your hand steady. So you use your, in my case, left hand to pull the test tube away from the loop. You flame it again to make sure there's no contaminants, and then you move the test tube to where the lid is. Notice I'm not moving my loop hand very much because, again, you don't want to risk flinging bacteria. Okay, so now you're done with your starting culture and you can set it off to the side. If we're pretending that I'm going to be inoculating an agar plate, we'll use my phone. You would lift the lid of the agar plate, keeping the lid at an angle to try and shield any contaminants. Then you would just streak the bacteria onto the agar plate, put the lid on it, set it down. And the last thing you need to do is sterilize your loop to eliminate any bacteria that might be on it. And then you can set your loop down and you're done. Okay, so it sounds pretty straightforward, but there's a lot to think about, especially considering there is fire in front of you. So we're gonna do that from the top one more time with a little less commentary from me. So again, you always want to start by kind of loosening the lids on your test tubes if that's what you're working with. So I'm going to have the test tube in one hand, my loop I'm holding like a pencil in the other. Always flame your loop first. Remove the lid of your test tube, holding it at an angle, pass it through the flame, stick your loop in, get some bacteria, keep your loop hands steady, don't move it if you can. Move the test tube away from your loop, pass it through the flame and cap it. You are now done with that. And then you get whatever it is that you're putting the bacteria in. If it's an agar plate, you're just going to lift the lid, streak the bacteria onto it, and put the lid back on your agar plate. If you're transferring into another test tube, you do the exact same process. Hold the new test tube at an angle, take the lid off, bring the bottle, or I'm sorry, the test tube to your loop. Again, you don't want to move your loop hand while there's bacteria on it. You'd swish around the loop so that you can transfer that bacteria. Pull the test tube away from your loop, flame it, cap it, set it off to the side. And then the last thing you always do is flame your loop again, so that way you can set it down and go about your business. Okay, so you always start and end with flaming your loop. Okay, I'm gonna go off camera now. All right, so I just walked through these points. The more specific steps in proper order, like I demonstrated for you, is on the aseptic transfer handout. So I just want to remind you some things that we talked about in the previous video, which is about the types of media. We're mainly going to be using agar plates and broth. So that's what I included on this PowerPoint. But do remember we have slants and deeps, both of which are solid and made into test tubes. Sorry, I have hiccups. So this top agar plate on the left is what a sterile agar plate looks like, okay? Notice there's no colonies or anything on it. Once you add bacteria to it, you're gonna put it in an incubator and you allow it to grow overnight. And you will see these colonies in the morning wherever you put bacterial cells. We always grow media. Whenever you put an agar plate into an incubator, you put it in upside down. That way the lid is facing the shelf of the incubator, because a lot of times moisture will form on the plate and you don't want it touching the actual solid media where your bacteria are, because it can contaminate it. It can cause them to the different colonies to like mix and stuff, and you don't want that. And so by putting the plate in upside down, any moisture that accumulates on the lid. All right, with broth media, again, you can see what it looks like when it's sterile. That's that top picture. And then once you put your test tubes into the incubator and allow them to grow, normally 24 to 48 hours, they will become turbid, which lets you know that the bacteria has grown, okay? But bacteria grow differently in types of media, okay, especially on agar plates, but there will be a whole lab about that. So let's look at how things might grow in the different types of media. So with a slant, okay, sometimes so on the left again is the sterile slant, there's nothing on it. And then the three pictures to the right show the different ways bacteria and organisms may grow on a plate. So the second picture, if you look real close, you can see the outline right over here of the bacteria. And then it's easier to see where there's bacterial growth on these other two pictures here. And with the deep, again, remember you use an inoculating needle. And so I mentioned to y'all earlier that you can look at motility, which is what's happening in this particular photo. And so when you stab a deep, you're only introducing bacteria along that stab line, right? Because they're on the tip of the needle, you're inserting that tip of the needle into the media. So you're putting the bacteria in a very specific place. Now, if the bacteria do not have any flagella, then they're stuck in that stab line and they can't grow anywhere else. And that's where you get the far right picture. Do you see how there's only growth in that stab line? Those are non-motile organisms. On this other picture, I'm going to go ahead and label it photo A. So we just talked about photo B, which were non-motile. But notice on photo A that the rest of the media kind of looks cloudy, right? And this stab line is not clear and concise. That's because these organisms are motile. And even though you introduce them in the stab line, they use their flagella to quote unquote swim away. Okay. Remember, the proper term for flagella is run and tumble. But that's again what deeps are used for. Now, when we look at broth cultures, for some reason, whenever people take broth cultures out of an incubator, they automatically want to not shake them hardcore, but shake them a little bit so they can see, some of the growth, see things swirling around. It is pretty cool. Can't entirely blame them for that, but you don't want to do that because organisms grow differently in broth, partially dependent on their oxygen requirements as well. So tube A and B, the bacteria are growing pretty evenly throughout the test tubes, okay? Now, if you look closely at B, it looks like there's little chunks of growth in there. So we call that flocculent growth. Okay, and this is all spelled out in this picture caption here. Flocculent growth is B, has those little chunks. But when the broth just looks nice and even, it's evenly cloudy, like tube A, we call that turbid. Specifically, it's normally called UFT for uniform fine turbidity. But for the purpose of this class, we are going to keep it more simple. And we're just going to say that both tubes A and B are turbid. Okay, there's growth all throughout the tube. Okay, so that's what we're going to say to describe the growth for tubes A and B. Now with tube C, the growth is only at the top. Okay, this is not my favorite picture because it looks like it's falling into the media, but normally there's growth only up here at the top, and that's called a pellicle growth, okay, pellicle. And then if we move down to D, where all the cells have gone to the bottom of the test tube, a lot of times these organisms don't like oxygen, so they're going down to the bottom where there's not as much oxygen. This is called sediment growth, okay, sediment growth. Okay. All right, that is the end of this video. Make sure to answer they're not really questions at the end of the aseptic transfer handout, but you have to look at the pictures. Actually, let me go ahead and show that real quick. Let me get out of this. Hold on a second, y'all. Here we go. Let's actually look at these real quick. So I'm sorry. this was just showing you if you were to aseptically transfer bacteria onto slant, what it can look like. So you already kind of saw a picture of this earlier when I went through the PowerPoint. And then the last thing in the results section is looking at 4 different organisms and how they grew. And so you want to make sure that you go through and answer on your own how each of them grew. Is it pellicle, sediment, or turbid? All right. I'll give you a hint. 2 of them are turbid. 1 is pellicle and 1 is sediment. So go ahead and right now, while you're listening to this, look at these pictures and make your guess. I'm going to count in my head to 10, and then I'm going to tell you the answers. Okay. All right, if you need more time, push pause, because you definitely want to make sure you understand these terms on your own. So with Bacillus subtilis, notice that the growth is at, the growth is at the top of the test tube. Okay, so that is pellicle. For Streptococcus pyogenes, the growth is at the bottom of the tube, so that's going to be sediment. And then for both E. coli and Staphylococcus aureus, there's growth throughout the tube, so those are going to be turbidity throughout. All right, now for real, y'all, that's the end of this video. Let me know if you have any questions.